Thank you very much. It's a pleasure to be here. It's a pleasure to be back in Madrid. I'd like to thank the organizers, of course, but you know what I'd really like to do is to thank those of you who made posters in this meeting. And I frequently go to meetings, and it's very painful to go to the posters because there's very little of interest, but there were so many wonderful posters in this meeting that I really enjoyed, I think, that aspect of the meeting even more than the presentation, so thank you for making those posters. Of course, we're here to pay homage to a very good friend, who I regard as one of the kindest, generous, most devoted friend that I have. And unfortunately, it does go back almost 40 years. It's a long time. But over those years, our families have become friends as well. And I wanted to show this one picture, which was taken about 15 years ago when I spent a summer here in Madrid with Inez. And here's Ross, Zach, and Manuel. Who do you think are my kids? Well, sometime after that, we were fortunate to have Manuel as our guest in San Francisco, and he stayed some months in our house. And despite the strenuous efforts of my wife to adopt Manuel and have him stay with us, he did escape, and he returned to Madrid. And in fact, I think he is here today. Is that true? Manuel, are you here? He was supposed to come. Okay. Well, I first met Inez and Peter when I went to do a postdoc in Cambridge, and I'm very pleased to be able to take this opportunity to thank both of them. Peter for introducing me. I came from graduate work doing enzymology of DNA replication in E. coli, knowing nothing about Drosophila genetics or development. And from Peter, I got a very good education in developmental embryology, and from Inez, who was teaching both Peter and me Drosophila genetics. Well, one of the things that I picked up, of course, while I was in Cambridge as a postdoc, was a study of engrailed, which you've heard about already this afternoon. And I studied engrailed for some years in my own laboratory, did a thorough genetic analysis of the gene. We cloned the gene. We showed that, in fact, engrailed is expressed specifically in the posterior compartment of the wing disc. We went on to isolate the protein. It's a homeodomain containing transcription factor, and we even crystallized that structure with its cognate DNA. In the course of all that work, I would say that we learned a lot about engrailed, but we didn't learn much about the biology in doing that. But knowing that it was a transcription factor, we then went on to look for targets of engrailed, and in that search, we identified and cloned hedgehog as a target of engrailed in the posterior compartment. Also identified cubitus interruptus, which is expressed in the anterior compartment completely. I'm sorry. I'll try to speak louder. Okay. As a target of hedgehog. And we studied how hedgehog works. We studied how cubitus interruptus works. And in a lesson that I learned from both Peter and Antonio, to conduct these kinds of studies at single-cell resolution, we were able to clearly establish that hedgehog is expressed specifically in the posterior compartment, but the protein moves to cells that don't express it. And we're able to do that because we established that at a single-cell resolution at the compartment border. But then the issue became, how does hedgehog move, either short distances or long? And that's the topic that's occupied us for these many years since then. And I'm going to answer for you today, not only for hedgehog, but also for DPP. And Ernesto, DPP does not diffuse across a disc. And I'm going to show you how it does move. And I want to emphasize that. It does not diffuse. It disperses. Diffusion is a mechanism of dispersion, and it does not move by diffusion. Okay. Okay, here is how it moves. And we've pictured that here, expressed in this blue cell here, and what I'm going to show you is, in fact, DPP and these other signaling proteins move at a distance by moving along or in these structures, which are specialized filopodia, which we call cytonemes. So that is the title for my talk, I'm sorry. Signaling at a distance, communicating by touch. Now, the Bible if you will, tells us that there are three modes of signaling for cell-cell communication. There's endocrine signaling, that clearly exists, okay? Hormones are secreted into the vasculature and they distribute rapidly throughout the body and establishing uniform concentrations. We all know about neuronal signaling, which imparts spatial, temporal, and quantitative specificity to the signal, even when the neurons are very far apart from their targets, by virtue of direct contact at synapses. But the Bible also tells us that there's a mechanism of paracrine signaling, which is spatially limited, whereby signals are secreted by cells into extracellular space, then to diffuse to somehow find their receptors somewhere. Okay. That's the mechanism that I'm going to discuss today. But what I'm going to say is, very briefly, because I know my time is short, that development has a very small but interesting vocabulary of signaling proteins. TGF-beta, or the fly DPP, FGF, hedgehog, Wnt or wingless, the Notch delta system, and EGF. It's small, it's universal, it's true in all tissues in the fly, in all tissues in virtually all animals, and I'm going to argue and try to get to showing you that the mechanism by which these paracrine signals move across space from producing to target cells is also universal, and it's at points of contact. Okay, so I'm going to skip this because I know my time is abbreviated, and simply take the time to introduce you to the system I'm going to describe. Our Drosophila wing imaginal disc includes not just the cells of the wing disc, but also a tracheal branch which directly associates with the wing disc and makes this tiny structure here, which we call an air sac primordium, because it goes on in the adult to generate the major tracheal structure of the adult, the dorsal air sacs, which provide oxygen to the flight muscles. But the cells that will produce those flight muscles are also in this region shown in here, where we've illuminated them with GFP, and you can see them here, although they're not visible here. So all of these three types of cells, the wing disc cells, the tracheal cells, which are going to make the dorsal air sacs, and the myoblasts, which are going to make the flight muscles, are together, and they're communicating. I'm going to show you how they do that. Okay, so that's here. So these cells will go on to make the dorsal air sacs, the flight muscles, and the structures of the thorax, the wing and the thorax. Okay, so in pictorial cartoon, we see, in this case, DPP is expressed in this stripe along the anterior-posterior compartment border. There are a few cells here in this nondescript region which express FGF, but the wing disc itself does not express the FGF receptor, but indicated here is this tracheal branch with our air sac primordium that will go on to produce the dorsal air sacs, which does express the FGF receptor, and it receives FGF from the disc, and that's shown here in cross-section. So this air sac primordium actually lies underneath the basal lamina, or the basement membrane of the disc, in direct contact with the disc columnar epithelial cells, which either, in this case, we're showing make either DPP or FGF. So simply to show that the air sac primordium here does respond to FGF with an antibody against DP-ERK, and here's a readout for DPP signaling indicated here also. Both FGF and DPP received from the disc. So here I show this picture simply to give you a sense of this air sac primordium, where we're expressing membrane-tethered GFP, and we can see that there are about 120 cells in this organ, which buds out through a process of tubulogenesis from this tube here in response to FGF and DPP. But if we take that same picture with that same CD8 GFP and overexpose it, we get a very different view of the air sac primordium, and that's shown here. It's not as pretty, but in fact, these filopodia or cytonemes extend out in all directions, and what I'm going to show you is those cytonemes, what we call them, are responsible for reaching out to the FGF-expressing cells in the wing disc, to the DPP-expressing cells in the wing disc, and taking up those proteins. And the signaling, the FGF signaling and the DPP signaling in that air sac primordium depends upon those contacts, depends upon the uptake at those points of contact. Okay. So the first question we ask, and I include this for Inez's benefit, is if we look at an earlier stage in air sac primordium development where it's growing out from this transverse connective, but it has not yet crossed over this line of DPP expressing cells, we can observe cytonemes extending out from the tip. Now, if we're going to argue that those cytonemes are involved in picking up those signaling proteins, FGF and DPP, they should have the receptor for them, and in fact they do. So what we did is we expressed both ectopically expressed thick vein, which is the receptor for DPP, fused to GFP an FGF receptor fused to cherry in those same cells to ask whether those filopodia, those cytonemes, would have the receptor in them. And they do, but the surprise was they're in different ones. So there are cytonemes that extend out from the tip that have the thick vein DPP receptor. There are different ones that have the FGF receptor. Who knew? We didn't know, but when you look, as Peter said, when you look, you see. Okay? So two different types of cytonemes. That is, the cell has segregated these receptors into these two different structures. If you simply look at the shape of those filopodia, you would never know. But this is saying they have two different functions. Their constituents are different. Now, in this case, what we're doing is we are expressing in our air sac primordium, we're just labeling the membranes with membrane-tethered cherry. That's why everything is red. Under DPP promoter, we're expressing DPP GFP. So this is a functional DPP to which we fused GFP. So this green stripe here, that's the wing disc. They are expressing DPP. The air sac primordium does not express DPP, but it's labeled with this membrane marker. And you can see these filopodia, these cytonemes extending down from the ASP to the DPP expressing cells. These clearly are picking up DPP from the wing disc, DPP GFP. These cytonemes out here do not. Why? Because they have the FGF receptor in them. They're immune to the DPP. They're reaching out to the FGF expressing cells out here, which I don't have time to show you, but what I do want to show you is a movie where the air sac primordium, again, is expressing the receptor for DPP, thick vein cherry, and DPP GFP is expressed in the wing disc. Whoops, sorry, let me go back. So you can follow these yellow puncta because they have both thick vein cherry and DPP in them from the DPP expressing cells here in the wing disc to the ASP. So we are actually imaging DPP moving from the wing disc producing cells to the target cells, to the receiving cells in the air sac primordium. So it appeared to us that these cytonemes that are picking up DPP are actually making direct contact, but how do we show that? We show that using this technique here called GRASP for GFP reconstitution at synaptic partners, which relies on the fact that GFP, this small protein, which has this active chromocenter that can fluoresce, can be expressed in the same cell as two separate peptides. Those peptides can find each other within a cell, fold to generate a fluorescent protein. But what Corey Bargman showed in her lab was that if those two parts of GFP were fused to a transmembrane domain, in this case CD4, and exposed on the exterior face of a cell, and if those two pieces were expressed on two different cells, if those cells come into close contact, for instance, at a synapse, a neuronal synapse, then those synapses will light up and be fluorescent, okay? We've asked the question, can we adapt this technique to our cytonemes to ask whether they make direct contact with the target cell? And in fact, they do. And I'll show you one example here where we've labeled the cytonemes themselves from the air sac primordium with a membrane-tethered cherry and expressed and one part of GFP, just the 11th strand of the 11-stranded beta barrel. And in the FGF expressing cells over here, we're expressing the other parts of GFP. And you can see that the tips, at least some of the tips, light up. So they're making direct contact at the same, essentially the same distance as a neuronal synapse, some 15 to 20 nanometers. And since we know that GFP does not fold instantaneously, it means these contacts have to be relatively stable. Okay. Moving quickly right along, if you're still with me. We've identified a number of genes which we can show are required to make cytonemes or to make functional cytonemes. I'm going to focus on one called capricious, which encodes an LRR, a leucine-rich repeat, cell adhesion single-pass membrane protein, sorry, Peter, that has been shown previously to localize to the tips of neuronal growth cone filopodia. And CAPS mutants have been shown to make defective synapses. So we've taken this sort of a candidate approach to ask whether we could identify genes that make proteins that localize to the tips of filopodia and ask whether they, in fact, localize to the tips of cytonemes and are they required. And in fact, if we express CAPS GFP in the air sac primordium, we can see that it's in cytonemes and it localizes to the tips. And in the absence of CAPS function, that is, if we express a dominant negative version of CAPS in the air sac primordium, you can see clearly it's an abnormal shape. The question for us is, in the absence of CAPS, do cytonyms, are they functional, and is there signaling? And what we can show, this is now the control, showing in cross-section here, here's our air sac primordium in red. The wing disc is down here where we're expressing DPP, GFP. Here's that stripe, okay? You can see the close juxtaposition of the DPP expressing cells in the disc and the air sac primordium. And actually in this transverse section, you can see it sort of sits down in a little cleft. Despite that proximity of the air sac primordium and the wing disc cells, the air sac is actually extending these short little cytonyms down in, and they actually touch the disc. And we can say that because if we use that grasp technique and we focus down from what I call the upper layer through down to the bottom, we can see that these lower layer cells make direct contact with the disc by virtue of this grasp fluorescence. But now we can ask, is that grasp fluorescence still present if we knock out CAPS, this protein that localizes to the tips of the cytonyms? And it isn't, and that's shown here. So again, CAPS is this protein which localizes to the tips of our cytonyms. This is the juxtaposition of the air sac primordium and the disc cells. Control, here's a frontal section showing the grasp, that is the GFP fluorescence that's generated at points of direct contact, either in a frontal section or a sagittal section. And if we express this CAPS dominant negative, gone, essentially gone. More importantly, DPP signaling in a control disc, I've shown you this before, where we have this DAD GFP readout, which tells us where cells are active in DPP signal transduction, completely absent if we express CAPS dominant negative. Mind you, we have not changed the genetic constitution of the wing disc cells that are making DPP. They are wild type. We are expressing CAPS dominant negative only in the tracheal cells in the air sac primordium. The wing disc develops normally. The wing disc doesn't care whether the air sac develops or not. We can make flies that have no dorsal air sacs. They don't fly very well. They don't fly very long, but for the exterior, the cuticle and the wings are normal. The wing develops normally. In fact, you see that here. The wing disc, these are wing disc cells that are responding to DPP when we block CAPS function just in the air sac primordium. So I want to emphasize this. What we can say is that signaling is dependent upon CAPS in the cytoneme-producing cells, that is the air sac primordium cells. Without CAPS, cytonemes don't form contacts. I showed you that with the grasp experiment. That is, signaling is contact dependent. That is, the only DPP that signals from the disc to the air sac primordium traffics by cytonemes. And if extracellular DPP exists, I can't say that it doesn't. What I can say is that if it exists in the absence of cytonemes, in the absence of direct contact, it doesn't signal. Okay. So with that, I'm going to turn to hedgehog. And I think what I'm going to do here is just refer to the beautiful work of Marcus Bischoff, who was a postdoc in Peter Lawrence's lab, also the work in Isabel Guerrero's lab. They've shown that hedgehog traffics by cytonemes, and they've characterized cytonemes that extend from the hedgehog-producing cells into the anterior compartment, carrying hedgehog to those anterior cells. We have a sort of a companion story showing that the anterior cells that receive hedgehog also send cytonemes into the posterior compartment, and they take up hedgehog from the posterior cells. So hedgehog is using both sending and receiving cytonemes. And I'll just mention this very briefly because it may be of some interest to some of you. This wing disc, which is showing us hedgehog GFP expression in the posterior compartment and patched cherry expression in the anterior compartment. Patched is, of course, the receptor for hedgehog. This is an unfixed disc which has two backs, one which encodes hedgehog GFP, one which encodes patched cherry. Both of these are fully functional. Each of those backs can rescue a null mutant to wild type, and yet we can image that hedgehog and that patch protein in an unfixed disk, and it's a very powerful system. And let me just show one image. So what we're doing here is looking at cytonemes that extend from the anterior compartment into the posterior compartment with this hedgehog GFP back. That's the green spots here. And the patched cherry back, that's the red spots here. These red spots are motile, and they're little vesicles that move within these cytonemes, which we're marking with this third marker here, which is a membrane-tethered infrared fluorescent protein. And you can see these patched vesicles moving within these cytonemes, some of which have hedgehog in them. Okay. Again, hedgehog moving from the producing cells into the receiving cells. Okay, I'm going to skip that. In the interest of time, I'm going to skip all of this. Well, this might be of interest. So here's our air sac primordium, the tracheal branch, and here are our myoblasts, which are actually all surrounding these tracheal cells. And we can show that the air sac primordium is extending out cytonemes with this grasp technique that are contacting the myoblasts, and that the myoblasts are also making cytonemes that extend to contact the air sac primordium. And we can show that the myoblasts make, so what I meant to say is that what I'm not going to have time to show you is that the disc expresses wingless. Wingless is signaling to the myoblasts to negatively regulate delta, and delta activates Notch in the, I shouldn't say disc, this should say air sac primordium. I apologize. We can show that the myoblasts are making cytonemes that have frizzled the wingless receptor in them, and we can follow wingless GFP expressed in the disc into these cytonemes together with frizzled. We can show that the myoblast signals to the air sac primordium via filopodia that contain delta, and that we can knock out those cytonemes, in this case with RNAi for diaphanous, which knocks out the cytonemes and knocks out Notch signaling in the air sac primordium. So our model here is that the wing is producing wingless, which signals via these frizzled containing cytonemes to the myoblast, that these same cells make cytonemes that extend to the air sac primordium that have delta that activate Notch signaling there. Okay. So in conclusion, this is our cytoneme direct transfer model. Cytonemes are filopodia that make direct contact with cells even at long distance. Cytonemes exchange signaling proteins at sites of contact and transport signaling proteins between cells. Diffusion, a mechanism of dispersion, may appear to be the simplest mechanism for dispersion, but experimental data that I've shown you today implicates cytoneme-mediated movement and exchange at sites of direct contact. And I would just ask you to ask yourselves, if you want to consider diffusion, is there any experimental evidence that supports it? I would argue no. Okay. Finally, basically we're saying that these cells, like neurons, make asymmetric extensions that send and receive signals. Every cell in that context is a neuron. And finally, those who are carrying on this battle in the lab, Felipe Andres Ramirez Weber, who discovered cytonemes some years ago, Makoto Sato, who discovered the air sac primordium and showed what it does, Frank, Sugata, Huang, and Wei Tao, who've continued that work. And thank you very much for your attention.